Right, welcome back. Welcome back to our keynote speakers this afternoon. So it's a huge pleasure to introduce to you this afternoon Susan Wessler, who's the Professor of Genetics at the University of California, Riverside. Susan is a member of the National Academy of Sciences. In fact, she's the Home Secretary. There's the first woman Home Secretary of the Academy. She's a member of the American Association for the Advancement of Sciences, the American Academy of Arts and Sciences too. And this year, she was awarded the FASEB Excellence in Science Award. And it's my understanding that it's particularly appropriate for her to be here to give a keynote speech to you today because of her meeting with Barbara McClintock and how that helped her to launch her own career in the understanding of transposable elements and their evolution and functions. So without further ado, Susan's going to give us a keynote speech on the success strategies of transposable elements that rapidly diversify genomes.
Okay. My voice is going, and hopefully it'll make it through this talk. Get this in my back pocket here. Okay. So as Chris said, it's a real special pleasure to be here at the meeting, but it's also a real special pleasure to be back to where I had met and was influenced by Barbara McClintock. And I thought if we could have a little lights up, maybe just the ones in the front down. Oh, maybe not. Okay. So I think that working on transposable elements for 30 years already, what you realize is that it's really the technology that allows you to ask and have answered deeper and deeper questions. And I think all of us, in a sense, need to go back to who were the first genomicists. And McClintock, as probably the best cited geneticist of her time, looked at genomes in a very different way. And in fact, it's through the breakage fusion bridge cycle that clarified a lot of the functioning of transposable elements. So what I want to say that she was, I think, if not the first, one of the first people to think of the genome as an entity more than just a collection of genes. And this is her quote from the Nobel lecture, which I'll let you read. So her influence, really, her main influence in terms of my work is in understanding the evolutionary impact of transposable elements. How do they impact and diversify genomes? And she, of course, was way ahead of the rest of us when she proposed her genome shock theory. And this theory basically says that transposable elements are components of all genomes, that they are maintained in an inactive state, that stress conditions may activate transposable elements, which I'm abbreviating as TEs here, and that transposable element movement increases the mutation frequency. Finally, that TE-induced mutations or rearrangements may be adaptive. So when I started working with transposable elements back in the early 80s, back in 1980, what I did was isolate genetically, clone the transposable elements, McClintock's AC and DS transposons. And I didn't appreciate then how valuable it was really to start with with mutations. And that is that if you have an unstable allele like these spotted corn kernels, it tells us that it tells one that they're working with an actively transposable element, an element that's actively transposing, and it also tells you where that element is in the genome. So from the isolation of those elements, we were able to identify what's called now class 2 DNA transposons, and I'm just going to give you a brief background. And this would be a transposable element family. It is composed of autonomous or transposase encoding elements, and then there are most of the elements in the genome, as we now know, are in fact non-autonomous elements. These elements do not code transposase, either because they are, many are deletion derivatives or defective, and some I'll talk about in a bit. But there, as I said, there are generally many non-autonomous elements in the genome and a few or no autonomous elements in the genome. So the way an element moves, unlike retroelements, is the transposase binds to the end of the elements in the family. It dimerizes the element and the transposase excise, insert elsewhere in the genome. This is why they're called cut and paste. But the way they increase their copy number generally is if this donor site is then filled in by gap repair or some other mechanism. And that way, every transposition can result in an increase in copy number. Genomes contain multiple transposable element families. And so, for example, they are their own regulatory system. So the transposase from this family can move the elements that have similar ends. The transposase in this family can move this, but this transposase doesn't affect that family. So after working with these genetically identified elements, we found that there were several problems, and that is if we were interested in in evolutionarily relevant elements. And that is that, in fact, most of these elements, most of these mutant alleles, led to the identification of what turned out to be very low copy number elements that are mutagenic, which is no surprise. They caused mutations. And really, these elements, we now realize, have a minimum impact on gene and genome evolution. Such elements as the P elements, A, C, D, S, SPM, they're hyperactive elements that sort of don't get it. So the question then is, where do we look for transposable elements that impact genome evolution? And this, as most of you know here, we now know that genomes are filled with transposons. For example, some human genes, and I've shown the exons of some gene at the top, and at the bottom all the elements that are in that gene, that some human genes and genes from other organisms have up to 100 transposable elements in their introns. However, virtually all of these elements are monomorphic. That is, most of the elements in my genome are exactly the same, in the same positions as the elements in your genome. So because of that, because of this sort of lack of activity, we can't determine how transposable elements alter gene expression or how transposable elements can actually attain such high copy number. How do they get to, like ALU, 30,000 copies in a burst without killing their host? And that was really the question we were interested in. So as I said, we wanted to identify the high copy number elements in plant genes. And so our strategy, and this is actually a region of the Borley chromosome, just to show how many transposable elements there can be. These green boxes are genes. This is from a colleague, Alan Schulman. These are mostly retrotransposons. So the idea is to find a species or group of species in the midst of genome expansion, to then identify and isolate an active high copy number element, and then analyze how it amplifies in the genome. Okay. Obviously, the second part is the tough one. Okay, so we had our group of organisms. Well, I was already working on maize, and over the years we've come to realize what a dynamic dynamic genomes this group of organisms has. Even though they evolved from a common ancestor only about 60 million years ago, they differ in size by 20, 30-fold. With rice, the smallest and this is wrong, it's 350 megabases, must be an old slide. Sorghum, twice that size. Maize, a little smaller than the human genome. Barley, twice the size. Wheat, even larger, and oat, off the scale. Polyploidy accounts for some of this, but most of it really is due to transposable element activity. And what I've shown here is that all the cereals really have about the same gene number, and that number is about 30,000. And what we find is that the distance between the genes vary. So these genomes, the grass genomes, are largely synthetic. And so you see that the rice genes, for the most part, here would be 3 genes. They're closer together in rice, further in maize, further in barley. This is hugely oversimplified, but it holds. So if we look even more closely, what we call the grass ecosystem is that we have genes that are separated by these massive clusters of 100 to 200 kb of retrotransposons. And this was discovered by a colleague of mine, Jeff Bennetson, many years ago. So I'm not going to be talking about retrotransposons. In fact, what I'm going to be talking about are the elements that were discovered in my lab almost 20 years ago, which is pretty scary, with Tom Bureau, who's now at McGill University. So we discovered these little elements called mites, and I'm going to tell you a little bit about that discovery. And so this started with a mutant allele of a gene that we were working on, Mays-Waxy gene. And what we found, that it had a small insertion, it was a stable mutation, and in fact was the first mite. And the way Tom discovered it was by doing, I don't think it was BLAST, you guys probably know a lot more about this than I do, because it was 1992. He did computational analysis. He looked at the wild-type genes, plant genes that had been submitted just as wild-type sequences. And what he found, that many of them had related elements that were in their introns and 5' and 3' flanking sequences. So these are normal genes, and these are just sequences that people had deposited, and it led him to discover this family of class 2 DNA transposons that are very, very abundant in plant genes. So I'm going to add them to the family here, that these are non-autonomous elements that we now know have amplified from a single element to very high copy numbers. And we're talking about copy numbers in the hundreds to thousands to tens of thousands, which is incredibly high for DNA transposons. So if we look at genomes, if we look at, say, the rice genome, or mostly any genome, mites, if you collect all the related mites and you generate a phylogenetic tree, what you see is this star phylogeny. And for those of you who are familiar with it, this indicates that mites amplified very rapidly from one or a few nearly identical copies, and then what happened is activity stopped and there was drift. So that's what this phylogeny indicates. If you look at genomes, what you see are lots and lots and lots of mite families that have burst, and the bursting is stopped. These are all essentially dead elements that are drifting. So what I'm going to do is tell you about sort of the life history of the one element that's been the focus of my lab, MPING. It's a high copy number transposable element. And for those of you who are my age or older, you'll recognize this. This is from a doctor show called Ben Casey. And it was in black and white. And it was started with a chalkboard, and they wrote man, woman, birth, death, infinity. It was very dramatic. So what I'm going to talk about is the birth, the growth, and the death, the presumed death of the MPING element. So I'm going to start with, and as I said, in order to understand how these elements diversify genomes and how they amplify without killing the host, we had to isolate an active one. We had to isolate one in this time frame when it was bursting. And to do that, I had to switch organisms from maize, which at the time did not have a genome sequence available, to rice, which had a complete genome sequence. So this was work done with my collaborator, Sean Eddy. Essentially what we did is we used, again, a computer-assisted search. We used recon, and in that way we compared the existing rice genome to itself. We identified gene families in that way, multi-copy families. We then manually curated these, and Ning Jiang, a graduate student in my lab, identified one element which looked like a mite element. It was short, it had inverted repeats, and most importantly, it had 15 nearly identical copies. Any new element, as most of you know, will be nearly identical, because when the element moves, the two copies start out identical. So as with anything that comes out of the computer, it has to be validated at the bench. So what Ning did is reached in the freezer and pulled out some cells, cell culture, which had been sitting in the freezer for 4 years for a different project. And she used a technique that we had modified in the lab called transpose on display. I'll just show you very quickly. If you just make a primer it's a AFLP-like technique. If you use a primer from the end of the element, you take genomic DNA, and all you need to look at is lanes 3 and 4. You see lane 3 is the genomic DNA. We cut up the DNA, we put on adapters, we amplify. So lane 3 is the DNA from the plant prior to cell culture, and lane 4 is the DNA from the plant after cell culture. We can cut out all these bands, we can amplify them, we can blast them against the rice sequence and find out all these new insertion sites validated in that way. The other sort of plug for completing a genome is that we were able to, so the question is, where did this element come from? And there was a single element in the sequence genome. This is this large ping element, which encodes transposase. It encodes actually 2 proteins that are both required. And I'm going to get back to that at the very end of the talk. So for now, I've sort of told you that MPing, that mites attain very high copy numbers, but the sequence genome just had 50 copies. So that's not a very high copy number. So it's a long story, but we managed to work with a group at Kyoto University, and they had identified strains that we found had over 600 copies of MPing. So these are 4 related strains, and the strains I'm going to be talking about. So basically, we look like we have gotten that burst. We have strains that have 50 copies, strains that have 600, even up to 1,000 copies of MPing. So this is just to show you how similar these strains are, other than the MPing insertion site. It's the same kind of transposon display. So here we're using a probe, a primer, from an LTR retrotransposon where there are about 1,500 copies in the genome. And when you use the genomic DNA from Nippon-Bari, the reference genome, and these high copy strains, they look pretty similar. But if you use the primer, the only difference in this experiment is the MPing primer. The genomic DNAs are exactly the same. If you use it to MPing, this is what you see. So MPing, Nippon-Bari has 50, and these guys have, as I said, up to 1,000 copies. So MPing has burst independently in these high copy number strains. The next question that was really of interest is this element still moving in these high copy number strains? And the way we did this experiment is really the only way that you can validate whether an element is truly transposing, and that is to see it right before your eyes. Polymorphism is not sufficient. So essentially what was done in this experiment was we had 10 siblings, 10 plants, isolated DNA, grew them up, did transposon display, took one plant, took the seed from this plant, looked at the next generation, and then finally took the seed from this last plant and looked at the next generation. And so what you see are the new insertions. So every generation, we're seeing an increase in copy numbers. So for example, you could see in this generation, in this plant, there's a new insertion, and the next generation, that insertion is segregating. And actually, that'll come into play later when we talk about identifying heterozygous and homozygous insertions. Okay, so what we find was that there are approximately 25 new insertions per plant per generation. There are up to 50 and 60 insertions in some plants. And the question we had, now we had our material, the question is, how does rice survive a burst like this? And basically, where do the MPing elements go that allow the plant to survive? These are perfectly healthy plants. Okay. So what we did, and this was 2 years ago, we used vectorette sequencing. We used 454, and we looked at about 2,000 insertion sites in a population of 24 of these plants. So essentially what we found, the first surprising thing, is that the element avoids exons. And we now know this is the case because we can look at the early events. The element avoids exons. And we think it's because of a target sequence preference. It prefers to go into AT-rich sequences, and rice exons are GC-rich. Okay? The second thing is, I think most interestingly, is it preferred to insert upstream of the transcription start site. So what we have here, this is the gene, and these are upstream regions, and you could see that there is a preference for within about 750 base pairs of the transcription start site. We've recently broadened this study to look at not just MPing, but to look at all of the mite elements in wheat, in brachypodium and other grasses, and essentially most mites have the same preference near the transcription start site. Okay. The other question we want to know, what is the impact of insertion on transcription? And this was done 2 years ago, and it's being done now, redone in many different ways with RNA-seq. This was done with microarrays, and essentially what we had was 710 alleles that had an MPing element in or near a rice gene. And then we had the alleles that did not from the sequenced Nippon-Bari reference genome. So we're basically just doing microarrays and comparing the expression and asking what is the impact of MPing insertion on these 710 genes. So essentially what we found, we looked at 31,000 plus genes, isolated RNA from the high copy strain in Nippon-Bari, and we found that the vast majority, almost 80% of the MPING insertions had no impact on gene transcription. Okay, that what we find in Nippon-Bari is equivalent to HEG4. And although when we first looked at this, I was pretty disappointed. I was sort of cut my teeth on these elements that really did some damage to the organism. And we saw lots and lots of differences. But the fact that this had no difference, that the differences were so subtle really was very telling. I mean, this is why this element can attain high copy numbers. This is a successful element. So the other thing is the majority of the other 156 alleles had increased gene transcription. About 2/3 of those showed higher. And when I say higher, just really tweaked, like 2-fold to 5-fold difference in transcription, not a major difference. And there were ones that had reduced expression, but very few. Okay. So the other thing that we're pursuing, and I'm going to talk about this a little later, very soon actually, is that some MPING alleles are now stress inducible. And the stress that they respond to is salt stress and cold stress. So I'm going to get back to that in a second. But I want to say that our collaborators at Kyoto University had another really valuable resource. And what they had done is they had taken one of these high copy number strains, EG4, get this right, and they had, and I think I have it here in the next one, they took one seed 20 years ago and they propagated a new line. So 20 years ago, one EG4 seed was used to start HEG4. Each line has been self-pollinated. Rice is an inbred. So it's a wonderful organism to look at these type of changes because it's relatively simple. The rest of the genome is simple. So the question really is, what can a burst of a transposable element do to diversify an organism in as short as 20 years? Okay? So what we've done, and this is part of a much larger project, is we've sequenced, and we're in the middle of assembling HEG4, and we've sequenced this so far to about 100, this is Illumina reads, sequenced to about 150-fold coverage. We're nearing the last stages of assembling this, and then we're sequencing the other strains, EG4, to lower coverage, aligning it to HEG4. And the initial results, and these are preliminary because the data is still being analyzed, is that when we compare these, so we're comparing here HEG4 to Nippon-Bari, the reference genome. We have 120,000 SNPs that distinguish them. When we compare it to EG4, we also have about 120,000 shared SNPs, but the private SNPs that these have are very few. So between these two, the difference between EG4 and HEG4 is less than 100 SNPs, and these are homozygous SNPs. So Sophia Robb, who joined the lab, actually this is a joint project with someone that some of you may know, Jason Steich, who's at Riverside, and Sophia is a shared postdoc. And so she has been developing software scripts to easily identify and align the MPING elements relative to the Nippon-Bari genome. So this is done rapidly. She actually did this because one of the things that I've done is I have a very large teaching program. It's a HHMI professor program. And it really is to replicate my research lab as an undergraduate classroom. And what we've done, and it's in its 4th year. I started it when I was at the University of Georgia, and it's been expanded at Riverside. It's a class for incoming freshmen. It is a computational and a wet lab, and the experiments basically come right out of my research lab. And so what Sophia has been doing is we have a class project, and this year the class project is actually to identify all the MPING insertion sites in one of these land races. So what she did is she developed this script so that we could then parse out each of the MPING insertions to the 12 rice chromosomes and assign the chromosomes to teams of students. So they're in the middle of analyzing that now. The other thing that we found is, the other thing that she's looked at is that the insertion sites, because this is such a dynamic genome, the insertion sites, remember there's 25 new insertions on average per plant per generation. So some of the insertions, most of them are homozygous, but some of them, as a sulfur, are heterozygous. So she's developed another script to genotype the insertion sites, and this is based on the number of two types of of sites. One are the spanners. Those go across the site, which is here, and the other are the flankers. So something that when you sequence, you get only flankers or homozygous, some where you sequence and you get 50-50 or heterozygous. And finally, what we see are the rare events. So in this case, we see we see many, many spanners and very, very few flankers. We also see the opposite. We could also see excision events. So essentially what we find is as the depth of coverage increases, more the rare somatic insertions can be identified, and we can identify somatic insertions in as low as 20-fold coverage. So plant transposable elements are not confined to the genome, like to the germline, as in other organisms. They transpose all over the place. And obviously, when we look at two individuals and look at the somatics, they're completely different. The other thing is that when we look at where the somatics are in the genome, the spectrum of insertions is really almost exactly the same as it is for the heterozygotes and homozygotes, indicating that there really isn't any selection that is removing these elements, that the element has really found a sweet spots in the genome. So this is an example. This is the preliminary results, where we find that if we look at HEG4 and EG4, we find that after 20 generations, they share approximately 344 insertions, and these are almost all homozygous. And when we look at their private insertions, we find that there's more heterozygosity to them because they're newer. So essentially, after 20 years, these elements differ by over 200 or 300 MPing insertion sites. So that's what it says here. Most of the polymorphisms that distinguish HEG4 and EG4 are MPing insertion sites and not SNPs. Okay. So basically, in a small population, the amplification of, so in like 10,000 plants, MPing generates tens of thousands of new alleles and allele combinations. And this is in a self-pollinating organism. This is an organism, there's no new DNA coming into this population. It is just rice, sperm and egg, that's it. The question, though, that we're interested in is really the really important one, is does this translate into phenotypic variation? And to address that question, what we're doing is we're working again with our collaborators at Kyoto University. The first thing, we're looking at recombinant inbred lines. So the two parents of the recombinant inbred lines, in this case, Nippon-Bari and HEG4, differ in several characteristics. They differ in height, time to flowering, some stress responses, and we're looking at a whole lot more. As I said, HEG4 has been resequenced. Nippon-Bari is completely sequenced. It's the reference genome. RNA-seq is underway. And so what's been done is over the past 10 or 15 years, our collaborators at Kyoto University have generated this recombinant inbred line through essentially 10 generations of selfing. So we now have 275 plants that we're now in the process of, I think I'll have that in the next slide. So what we're doing is we're assaying the RILs for morphological traits and stress responses. We're sequencing we plan to sequence all 275 recombinant inbred lines. And the goal is to correlate MPing insertions with phenotypes, with candidate genes, and then to go ahead and validate the candidate genes by transformation. So that's a long-term project. Our collaborators are at the bottom. We grow our rice in Arkansas, because thank God I don't live in a place where it's hot and humid anymore. Our collaborators also are Tom Brutinel and Chi Sun. So was that time or no? Oh, I just thought I heard beep. All right. So this is a plea for any of you out there that are interested in this project, the 275 RILs, and assembling them and correlating the various MPing insertion sites or SNPs or what have you with phenotypic change. All right, so now we're going to get into more speculative stuff at the end. And what I want to talk about is how this system started, the birth, and that how we think it's going to die. So I mentioned at the beginning, or near the beginning, that MPing is a deletion derivative of the element ping. Ping, there's only a single copy of ping in the sequence genome. So we were very surprised for this, the surprise part of this experiment I didn't tell you, and that is that when the cell culture that gave rise to this, this DNA essentially doesn't have ping. There's no ping in the genome here. So essentially, no ping in the cell culture where MPing moves. So ping transposase is not catalyzing MPing transposition. So the question is, what is the transposase source? And what we did is we went back to the genome. And ping is the member of a superfamily called PIF harbinger. It's not really important, just to say that in the rice genome, and this was a paper that had done many years ago in the lab, we had characterized all the members of the PIF harbinger family in Nippon-bari. And so ping is where you see it. It's a one element over there. What we noticed is that there were another element. There were 5 copies of another element, which we called pong, obviously. These were virtually identical elements. So our question was, maybe if ping wasn't in the particular genome that we were looking at, was pong capable of moving mping? So the first question was, since pong, as I said, there's 5 copies, and they're in virtually all rice genomes. I'll show you that in a second. But because it's different from ping, we were able to make a primer that was unique to pong amplicons. And essentially we showed, first of all, that yes, pong is actually moving in cell culture, in this exact cell culture. Okay. The next thing we did was we made transgenic Arabidopsis plants. So these are tDNA inserts of just GFP with mping. We took the native rice pong element, the native ping element, and essentially we found that yes, we could get transposition of mping in response to either pong or ping. Okay. The other thing we did, we also set up yeast transposition assays. And we found that both in Arabidopsis and especially in yeast, the pong proteins are over 10-fold better than the ping proteins at mobilizing mping. Okay. So what I wanted to show you here is that this is the old-fashioned southern blot. If we use this is rice strains, a whole variety of rice strains, and if we probe with we use a ping-specific probe, what you see is that there's some strains have, I said before, Nippon-bari had 1 copy, some strains have a few more, some have none. There are no copies. Whereas pong is, as I said before, present in virtually all strains, and about 5 copies, and it doesn't appear to have moved. So what we concluded from this, and other data has supported it, is that pong is silenced in virtually all rice strains. And it's very, very silent. So here's an element that is incredibly active, or potentially active, I should say. And yet over all of the divergence of these rice strains, it hasn't moved. And plus it doesn't move mping in normal rice strains. Okay, so what I'm going to do is end with a model for both the birth and the death of mping that sort of brings a lot of this data together. So the idea here is that, remember, I showed you a phylogenetic tree of all the different PIF harbinger elements in the genome, and they arose sometime way back from some common ancestor. They diverged. So I'm going to focus in on just ping and pong. The other elements in the genome have inactivating mutations. So these are really the only 2 active elements left in the genome from this superfamily. Okay, so the model, what we say, what I have here, is that we hypothesize that at some point a pong element became awakened. It no longer restrained by epigenetic regulatory mechanisms. And this is not too hard to imagine if every rice strain has pong. And they're all capable of being active. So you can imagine that, hot weather, something or other triggered pong activity. And what happened is we hypothesized that it then led to the amplification of mping. And this is how the higher, and just mping, not to the other copies. Okay? Then silencing was restored. And so what I have here is this is the situation in most rice strains. Pong is silenced, and the 1 copy of ping is slightly active. Ping is a WIMP. It's a really, really weak element. Okay? And we've shown that in many, many different ways. So this is what we have. So the Nippon-bari, the sequence genome, has this. And 1 every 40 or 50 or 60 or 70 plants have a new insertion of mping. So it's very, very weak. All right. So what I hypothesize is that the transient loss of pong silencing leads to the first burst of mping before silencing is reinstated. Okay. Now we get to the next step. What we hypothesize again is that there was another transient loss of ping. This is to create the high copy number strains, the 4 strains we have. So what we're hypothesizing is that the transient the pong activity promoted not just the activity of not just the movement of mping, but also an increase in copy number of ping. And this is key, because this makes the system now Pong independent. So Pong has been epigenetically silenced, and then what happens is Ping now, because it has several copies of the transposase gene, can be on its own. It's free now. What I have here, the transient loss of Pong silencing leads to the amplification of both M-Ping and Ping elements, and that the increased Ping copy number can now sustain the burst without Pong. And so this is the current situation in the high copy number strains. So now the question is, how is this going to end? And I think it's pretty easy to see. I mean, usually, I hate to say that because what I've learned in science is what's easy for us to see, it never really happens that way. So I hypothesize that death is going to be rapid. And so what we have are these high copy number strains with 7 to up to 11 Ping elements, and this number is increasing. Okay, so as I said, they keep pumping out more M-Pings, and as a slow rate Ping, his copy number is increasing. So the increased Ping copy number increases the chance of Ping silencing, essentially. So it's only a matter of time before Ping inserts into a locus, will initiate antisense RNA, and this burst will end, hypothesize, if Ping is silenced and or when insertions harm or kill the host. So that's how I think it's going to end. And I'll end this talk by another quote by McClintock, again, sort of, in a sense, thinking forward, and that we know about the components of the genome that could be made available for restructuring, like M-Ping. We know nothing, however, about how the cell senses danger and instigates responses to it that are truly remarkable. And I wanted to thank my collaborators. My students, the students who are current in the lab are highlighted. My collaborators are over here, faculty collaborators, and I'd be happy to take questions.
Questions? Susan.
So recently it was shown that P elements, which were thought to also insert into transcription start sites, actually home to origins of replications. Do you know if your transposable elements home to origins? Or?
No, and I don't know how much is known about origins of replication in plants. So it might, but I don't know, right?
Yeah, what I'm struck by is the fact that you have all these different rice strains that have these 5 copies of Pong, and they all seem fine. What I'm wondering is whether the strains are just very close to each other, and that explains the similarity.
I missed that part there. Can you, the phrase you just said I didn't hear.
Sorry, yeah, I'm wondering what explains that similarity of all these strains for the presence of these 5 copies of Pong, whether it's just close evolutionary distance between them, or whether there's some sort of selection effect or something else that explains why these Pongs are still there.
Why they're still there? Is that
why they are shared stably between several rice strains?
Well, they're inactive, and I don't think they've been there for millions of years. So we're still looking at things that are fairly recent. So yeah, I think that would be the only thing, that these aren't that ancient, right?
Okay, question over here.
So why do you think that the non-autonomous Pongs didn't take off?
We tested that. So I think that the non-autonomous, I think that there's something special about mites, about the elements that can jack up their copy numbers. And it's probably something that's biochemical, that they're good substrates. And so we've actually tested M-Pong. We made M-Pong elements, and they don't move very well with Pong. I think that part of it is that the element, what's the word I'm trying to say? Elements co-evolve with their ends, okay? And they co-evolve so that they're less than optimal. That's why I think Ping doesn't move M-Ping very well, and it doesn't move itself very well, but that Pong, it's a cross-mobilization that it's restrained, its sequence that's evolved at the ends doesn't work very well with its transposase, but it can, under certain circumstances, if you get a non-autonomous from one family and an autonomous with another, I think that's how most mites are how they come about, right? The short answer is I don't know.
There's a question over in the middle there.
Yes.
That was a beautiful talk. About 80% you said of the ping insertions don't affect gene expression. What's the difference between those that don't and those that do?
We don't know. I think that just we don't know. Yeah. I don't know. I mean, I wish we'll know more. I think that some of them that don't affect gene expression will under stress conditions. So the 80% is under just normal growth conditions. So what we're doing now is we're really doing an extensive, RNA-seq analysis with a whole variety of pathogens, of various, physiological changes. Is it epigenetic? No, in fact, most of the MPING elements in the genome are highly methylated. Highly, highly, highly methylated. And it's not surprising because MPING is in transcription units, and it gets transcribed, and it guides, you get antisense RNA, it guides organoid complexes to methylate DNA as happens in plant genomes. So no, and it doesn't appear to affect the flanking sequences. So the methylation doesn't bleed out, and that's work from other labs who've shown that. And I think significantly, all of the mites that we've seen have none of the coding sequences in them. So MPING only has ENs, because if they did have the coding sequences, they would presumably silence the ping element, the big element. So they're sort of flying under the radar. Right?
Well, please join me in thanking Susan once again for a fantastic talk.